Welcome to the Australian BioCommons webinar series. My name is Christina Hall and I'm the host for today. So today's webinar has been developed by Australian BioCommons, the Sydney Informatics Hub and QCIF as part of the BioCommons Workflow Commons project. It's part of a series of events where we'll share useful information about the latest digital techniques, data and tools for the life sciences community. You can keep up to date with the latest Australian BioCommons news and events via the channels that we have on our screen. So before we begin, we'd like to take a moment to acknowledge the traditional owners and their custodianship of the lands on which we meet today. In my case, this is the Ngunnawal people. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. And we also recognise their valuable contributions to Australian and global society. So today we're very excited to welcome Dr. Michael Geaghan and Dr. Sarah Williams to the webinar. Michael and Sarah are both senior bioinformaticians at SIH and QCIF respectively. They specialise in single cell and spatial omics and support the wider research community by building reproducible bioinformatics workflows. So welcome to the webinar, Michael and Sarah, and I'm going to hand over to you now to get us started with spatial omics.
Good afternoon, everyone, and welcome. Welcome to our webinar on getting started with spatial omics. Today's webinar is going to be an introductory overview of spatial omics technologies, how they work, and how we analyze this kind of data. It's intended as a general introduction to a two-day workshop that we're hosting at the end of October, where we'll be guiding participants through hands-on spatial omics data analysis. For today's webinar, it's going to be divided up into two sections. First, I'll be giving an overview of the current state of spatial omics, including what spatial omics is, the different technologies that currently exist, how they work, and how to choose the right technology for your experiment. In part two, Sarah will then guide you through the process of how spatial omics data is analyzed, including an overview of the of the tools and analysis ecosystems that have arisen around spatial data, the general data analysis process, some of the statistics involved, and the unique challenges that spatial data poses. So, first of all, what is spatial omics? Well, essentially it is the quantification of biomolecular signals, primarily gene and protein expression, within a tissue and, crucially, while retaining spatial information. So it asks the question where in a tissue is a gene or a protein expressed. So in this diagram here, we have a tissue that we might like to study, the brain, and we might like to know what genes are being expressed between two different brain regions, or perhaps we'd like to know how gene expression is differing within a brain region, but between two different treatment conditions. Current spatial omics technologies allow us to take a whole brain slice like this and detect what genes are being expressed across that whole tissue and pinpoint exactly where in the tissue they're being expressed. Furthermore, using the expression profile that we generate from this process, we can do things like cell clustering and cell type analysis to determine what types of cells are present in each brain region and in what proportions. And this is an example of some of the data you might get from a spatial experiment. You can see the same general outline of a mouse brain section here, but it's now represented as thousands of these colored dots, where each dot is a single cell that's been segmented from that image, and the color identifies what cluster of cells it belongs to based on its expression profile. And I'll also take a minute to note here that while transcriptomics and proteomics are the major use cases for spatial technologies, and because of that, we will be often referring to transcripts and genes and proteins in this webinar. Keep in mind that there are also emerging spatial workflows for studying things like chromatin accessibility and other epigenomic signals, and those omics can be substituted in as well. So let's compare spatial to some of the other omics technologies that are out there. First, we have bulk RNA sequencing. This is a quite well-established method at this point, and it involves taking a sizable section of tissue and homogenizing or dissociating the cells within that tissue section before processing it into a sequencing library, performing sequencing, and doing data analysis. Because of that cell dissociation step, you're destroying all the spatial and cellular information in that tissue. And at the end of when you do your data analysis, you get an averaged or blurry expression profile of that entire tissue section. Now that process can be refined a little with cell sorting prior to dissociation of your cells, and that allows you to study, say, specific cell types within the tissue, but you're still getting that average expression of that cell type. However, bulk RNA-seq is still very suitable for many use cases and is a great choice for when you need a broad expression profile of a tissue, and it's also very cost-effective and covers the entire transcriptome. So it's great for profiling lots of samples and for doing exploratory analysis, but for more complex questions like how cell types are interacting with one another in a tissue, you just can't answer that question very well with bulk RNA sequencing. With the development of single-cell technologies, we could start capturing individual cells and barcoding them with DNA sequences. Thereby, when we generate our sequencing library and perform sequencing, we can later tie those reads back to individual cells. That gives us a much more fine-grained information about the cellular makeup of the tissue, and we can inspect things like cell types and subtle expression changes between cells of the same type. But we're still relying on a dissociation step, and so this is destroying all that spatial information. Spatial technologies, on the other hand, don't rely on cell dissociation. They involve processing intact tissue on, say, glass slides, and thereby maintaining spatial information about the tissue. This combines that high throughput and fine-grained analysis that we expect from single-cell technologies with the ability to localize gene and protein expression within a tissue, similar to how we can do with basic microscopy techniques like immunohistochemistry. Now, there are two main branches of spatial omics technologies. There's sequencing-based and there's also imaging-based technologies. We'll discuss these shortly, but the end result is that from both technologies is you get a per cell expression profile along with X and Y coordinates of where they originated in the original tissue. But first, when should you actually choose a spatial omics technology in the first place? It is important to remember that the current capabilities, limitations, and costs of spatial omics means that it's not always the ideal solution for an experiment. If spatial information is not crucial to your experimental question, you should be asking whether or not simpler or more cost-effective methods would suffice. So, as I've mentioned already, bulk RNA-seq as well as microarrays, they're still very valuable, especially when needing to assay lots of samples because they're faster and much cheaper than other methods. Meanwhile, single-cell technologies, while they are more expensive than microarrays and bulk RNA sequencing, they can get you that single-cell resolution data, and they're also typically giving you greater depth of sequencing than what you can get with current spatial technologies. But once again, if you're interested in things like how cells or tissues interact with one another, or you expect there to be, for genes and proteins to be differentially expressed in different tissue regions, and you need some single cell or near single cell resolution, then this is where spatial omics technologies may be the best option. As an example, two prime use cases of spatial omics technologies are in neurobiology and cancer biology. In neurobiology, you have the brain, which is a very complex organ composed of a wide variety of tissue structures and cell types that are interacting with one another in lots of complex ways. And spatial omics is well suited to studying this kind of complex tissue. This is an example from a paper from 2023. The authors used the stereo-seq sequencing-based method in this case. And you can see that they've taken an entire brain and sliced it into lots of thin slices and performed spatial omics profiling on each of them. And And with that, they've been able to identify the individual layers of the brain cortex just by their expression profiles. On the side of cancer biology, you also have another complex tissue, the tumor microenvironment. You have tumor tissue, healthy tissue, and various immune cells all interacting with one another here. And understanding those complex interactions is vital for understanding things like how tumors invade other tissue and how they grow, how the immune system is evaded by the tumor, and how we can combat those processes more effectively. This is an example from a 2024 paper using the 10x Xenium imaging platform. And here you can see that there are immune cells which are identified by various gene markers, color-coded with teal, orange, yellow, green, and red markers here. And they're interacting with the surrounding tumor tissue in purple and pink. And in the inset here, in the zoomed in image, you can see that because this is an image-based method, the individual cells have been, the authors have been able to segment the image into individual cells and pinpoint exactly where in those cells and which cells those genes are being expressed within. Again, it's by preserving that information about where in the tissue those genes are being expressed, the spatial technologies can help us better understand things like the tumor microenvironment in a way that single cell and bulk RNA sequencing technologies simply can't. So ultimately, spatial omics technologies excel when you really need to examine things like the localization of cell types within a tissue or the patterns of gene expression within a tissue and with as close to single cell resolution as possible. On the other hand, older technologies like bulk RNA-seq, they still do excel when you need well-powered, high sample size case control style experiments. And single cell technologies are great for when you need to study the differences between cell types within a tissue in an unbiased manner, but you don't care so much about where in the tissue those cells originated from. By the way, this table, this summarizes the use cases of where each of these methods excels, with green ticks and red crosses, respectively, and also where they're not as where they're somewhat limited for the application, which is what's indicated by these yellow diamonds. And just note that these slides are going to be available after the webinar, so you can refer back to these later. Okay, so let's discuss the spatial technologies that exist at the moment. As I mentioned before, there are two main branches. There's sequencing-based methods and imaging-based. They have their own strengths and weaknesses, and that's primarily centered around a trade-off between the spatial resolution and the transcriptional or proteomic resolution that they can attain. So this slide here gives a brief summary of those two branches, and we will come back to this in a few minutes over the next few slides. So for sequencing-based spatial technologies, we are primarily using something called a spot-based method. And this is where the tissue of interest is placed on a special glass slide that contains thousands of microscopic dots of barcoded DNA. The tissue on top is permeabilized, and the RNA or protein inside is allowed to hybridize with the spots directly beneath them. And once processed, that sequencing library will then contain barcoded transcript fragments that can be sequenced and linked back to their original positions on the slide, thereby retaining that spatial information. During analysis, this is often further paired with high-resolution microscopy images of the tissue slide, often bright field H&E stains, or alternatively immunofluorescent imaging. The most prominent offering for spot-based methods is the 10x Visium platform, although the company Stomix also offers a spot-based method called StereoSeq, which I've mentioned earlier. These methods work with both fresh frozen tissue as well as fixed or FFPE tissue. For fresh frozen tissue, the DNA spots on the slides have these poly-T sequences, which help to capture polyadenylated mRNA molecules. On the other hand, for fixed tissue, The spots will contain a capture sequence, a special capture sequence, and the tissue is additionally treated with a set of probes that can bind to specific known sequences within the transcriptome. It's those probes that then hybridize with the capture sequences on the slides, and and then that's what's sequenced and tied back to the original positions on the slides. Now, as a consequence of the probe-based design, fixed tissue can only be analyzed for the expression of predefined sets of transcripts. So for example, the Visium platform currently only supports human and mouse transcriptomes for fixed tissue, although they do cover the entire transcriptome. On the other hand, the poly-A capture method that's used with fresh frozen tissue allows for potentially any species to be analyzed. And it's also not limited to predefined sets of transcripts, meaning that novel transcripts can potentially be detected with that method. Now, while the spot-based methodology allows you to capture the full transcriptome of the sample, the main limitation is that the spots don't necessarily map one-to-one to cells. So when these methods first appeared, the spots on the slides were quite large and they covered multiple cells. So it's not truly single-cell technology. Now, as the technologies have improved, those spots have gotten a lot smaller to the point where they are now at the scale of single cells, but still be aware that this is not truly single cell because those spots can happen to overlap, say, the boundary of 2 cells or more. So it's not truly single cell. Now, another sequencing-based method that is somewhat unique and worth mentioning here is the geomics platform from Bruker. This is, they call it a digital spatial profiler. It's not spot-based, but rather based on regions of interest in your tissue. It's also probe-based, so it only works on fixed tissue. The tissue is typically stained with immunofluorescent tags so that you can visually identify specific cell types or cell boundaries in your tissue, and then you can select multiple regions of interest within the tissue that you want to sequence. Each region of interest needs to contain multiple cells, typically dozens or hundreds, so it is not single cell, but it is incredibly flexible due to each region of interest being manually selected and arbitrary in size and shape. In this sense, it's something of a hybrid between spatial and bulk RNA sequencing, and it's particularly useful if you want to study specific regions or cell types in your tissue and want to cover the entire transcriptome. It also supports proteomic analysis. Of course, this does require that you have a specific region of interest or cell type of interest in mind prior to running your experiment, so it's not always the optimal solution for, say, exploratory analysis. Okay, so that's the sequencing-based methods, and while they have had incredible success, they're still unable to achieve true single-cell resolution. So recently, imaging-based methods have arisen, which utilize fluorescence in situ hybridization, or FISH. This is a method used to fluorescently tag and identify molecules within a cell and visually locate them using fluorescence microscopy. Both RNA and protein can be quantified using this method, using tagged probes and antibodies, respectively. Now, in order to cover a wide range of transcripts, imaging-based spatial omics typically go through several rounds of probe hybridization, imaging, and washing. The probes used within each round are not specific to single transcripts. Rather, they bind to smaller sequences common to multiple transcripts. Instead, each transcript is associated with a unique set of probes, and it's through multiple cycles of probe hybridization, imaging, and washing that a particular sequence of positive and negative signals will build up in a given region of the tissue, and that sequence can be decoded and used to identify what specific transcript was present at that location. In combination with fluorescent dyes to stain nuclei and cell boundaries, the images can then be segmented into cells, and that allows each transcript to be mapped back to a given cell. The major platforms that support image-based methods are 10x's Xenium, Bruker's CosMx platform, and VizGen's Merfish platform. Each one is now capable of identifying hundreds to thousands of transcripts with subcellular resolution. And these platforms have several advantages, primarily that they obtain true single-cell transcriptional and/or proteomic information while retaining spatial information. They also don't require any sequencing. Instead, the spatial, transcriptomic, and proteomic information is all contained within the images that are captured by the machines, and all image data is processed on board. However, they do still have a few limitations. They still use a probe-based design, so they can only target predefined sets of genes and proteins. And additionally, they only support a few hundred to a few thousand genes at once. There's currently no full transcriptome solution on the market. And another limitation is that they do often produce quite sparse data. So that means you get low RNA counts and low numbers of genes per cell, which can be a hassle to analyze and interpret. So the best targets are highly and consistently expressed genes. So to summarize here, the sequencing-based methods are still not quite capable of reaching true single-cell resolution, but they're very useful for profiling the entire transcriptome, particularly if you have fresh frozen tissue that can be used with a poly-A capture method. This makes sequencing and spot-based methods very useful for unbiased exploratory analysis and hypothesis generation. On the other hand, imaging methods do reach true single-cell resolution, but they suffer from more sparse data and targeted gene and protein sets, making them more useful for specific hypothesis testing rather than exploratory analysis. So deciding on the right technology for your experiment really comes down to a few key questions. First, what's your spatial hypothesis? What spatial information do you really require to answer your experimental question? And is spatial data really necessary, or can single cell or even an older technology like bulk RNA sequencing be sufficient? And how high throughput does the experiment need to be? Do you need to cover the entire transcriptome, in which case you might want to use something like a sequencing-based method? Do you only need to sequence a few hundred specific genes, in which case imaging-based methods might suffice? Or do you only need a handful of genes, in which case maybe targeted microscopy techniques are more efficient? Another important question is what level of cellular resolution is required? Do you need true single-cell resolution that imaging-based methods can provide, or can spotted spatial or the geomics region of interest method work for your experiment? And perhaps most importantly, what's your budget? Generally speaking, you're always going to be balancing the cost of your experiment with the statistical power it can achieve. Spatial technologies, especially imaging-based, can be quite costly compared to other methods, so you will typically have fewer samples to work with, and that can make finding subtle expression changes quite challenging. If you need lots of biological replicates for your experiment, spotted spatial technologies or non-spatial technologies might be the better approach. Assuming, however, that you have settled on a spatial technology for your experiment, you also need to consider what platform is best for you. The choice is a little more nuanced and will come down to factors like the type of tissue you have available, the species you're looking at, and the transcriptional or proteomic coverage you need. And once again, your required sample size and budget are also a factor. We've adapted this table from a paper published earlier this year by Lim et al. Again, this will be available after the webinar, but it summarizes the various available technologies, at least to date. For spot-based technologies, Stomix StereoSeq has become quite competitive with 10x's Visium, and it offers similar or better cost for depth while also offering larger tissue sizes and near single-cell resolution. But it does only support RNA and fresh frozen tissue at the moment, so while Visium supports fixed tissue and can support protein assays. On the imaging side of things, Xenium has the better price point, although both Merfish and Cosmix additionally support protein assays. So, in summary for part 1, the goal of spatial omics is to provide single-cell or near single-cell transcriptomic and proteomic resolution, while crucially also retaining spatial information. It's very valuable for studying things like tumor microenvironments, interactions between cell types, and spatial gene expression patterns. Two main classes of spatial technologies are sequencing-based or spot-based and the imaging-based methods. They have their unique strengths and weaknesses primarily centered around that trade-off between your transcriptomic and proteomic coverage and how close to single-cell resolution you can get. And finally, there are several platforms available, and the choice of which one to use will typically come down to things like your budget, your required sample size, whether you have fixed or frozen tissue, whether your samples are from human, mouse, or some other species, and how much of the transcriptome, proteome you need to cover. With that, I'll hand over to Sarah, and she'll step you through the spatial omics data analysis process.
Yeah, so Michael's just given us a great overview of what spatial omics is and how it fits in with the other omics technologies and why you would use it and when you would use it. So what we're going to talk about in the second half here is sort of how to do the analysis, sort of the core workflow for doing this, these kind of analyses. And what I mean by that is, how do we get to whatever you get that comes off the machine, whichever technology you've chosen, or spatial technology, how do you get to this point where, oh, I know what, I've got a bunch of basal epithelia over here, and I can then calculate their distance to, plasma cells, or, consider the expression of different genes, and sort of get to this sort of point where you can really mine biological questions out of your data and sort of keep querying it for whatever end that you actually did the experiment for in the first place. So I'm going to walk through this workflow here, sort of spatial omics workflow. So there are a lot of steps that tend to be common to just about most, if not all, spatial omics experiments. And so, you will start with something where if you're working with, spots or cell-based technologies, you can, you've got to figure out how many copies you've got each gene in a cell or a spot, and you build this sort of big table called a counts matrix of what's where. And that's very dependent on the technology you're working with. You would do some QC on that. And as you go along, you do further steps, which you start to consider biology a bit more. So after you've done some QC, you then go and normalize your data and produce some reduced dimensionality, dimensional reduction representation. And if you've looked at the some single cell or spatial omics papers, you generally see these beautiful UMAP views with a bunch of spots with all of the cells. Producing that kind of visualization there, because that lets you then go on to cluster your cell types, figure out, oh, where are my B cells? Oh, these are B cells, and sort of really annotate what's going on in your sample. And sort of when you get to that point, then you can start to think about like the really exciting stuff, like the hypothesis testing, or the comparing group A versus group B, and all of that kind of fun stuff. Your options really up here in these first few steps are pretty common, but once you get to that downstream analysis step, you just options explode, and you've got a lot of opportunity for mining your data sets. And I'll just flag here that this is an iterative process. You really can't get a script that'll just run you through the whole process in one step, because there's a lot of decisions to be made across at each stage. All right, so we'll walk through these steps in a little bit more detail. So talking about building that first step where you're building a counts matrix, this really is entirely dependent on what technology you're using. So in the case of a spot-based technology, it's actually quite simple to do. So here's that image from the visualization of the 10x Visium technology, where each read comes off with a read that identifies what the gene is and a spatial barcode that identifies which spatial spot it came from. You run a tool, it outputs a matrix for you. In practical terms, that's quite easy to do. And image-based technologies, to get to that point, there's a little bit more input required, because you don't have spots. You have to define the cells as your first step. So cell segmentation. And once you produce this sort of, you take your fluorescence, your images, and figure out where each cell is with a segmentation method, and then you can count the genes that fall, or transcripts or proteins or any other tech you're working with. I'm just going to say genes because most of these technologies, most of it's around RNA, but the same principles do apply to the other techs. Yeah, count that. Yeah, there generally does need to be tweaked a little bit for your experiment because different tissues will look very different, and you will need to tweak some parameters to get the segmentation to work the best. And yeah, also your fluorescence staining can vary between experiments as well. So, but the tools are there to do that kind of work. And once you get there, once you're happy with what your cell outlines are, the next step is this building this counts matrix, where you run some tool to count where each transcript falls into each individual cell outline here. So this one here has 3 copies of the NRN1 transcript, 2 of GAD1, and 1 of pValb. And you can see that they're quite spread out. So there's some outside of your transcripts, outside of your gene regions. That's normal. And yeah, so getting this step right is important for your analysis. And once you've got to that point, you'll notice things like, oh, that one doesn't have very many counts in it. When Michael said that this data can be quite sparse. I mean, this is just 5 genes, but you do end up with cases where there aren't many counts in a particular, aren't many transcripts measured in a particular gene. And what we tend to do is to do like a filtering out, because if you've only got 10 counts in a particular cell, it's very hard to say anything about that cell if 10% of it is a particular gene. In reality, probably isn't 10% of the transcripts or proteins in that gene, a cell, sorry. Yeah. But one of the things, because we're working with spatial data, you've got to remember that can't be too strict with the filtering. So every time we take out a cell, we then have this gap. So there's this balance between, on the filtering step, where we want to keep cells so we know where they all are relative to each other, but some of them are very difficult to analyze just through counts. Yeah, and this is one of those things where you have to sort of look at your data and decide, and okay, well, I need to be a bit more gentle with this one, or I can be more, do more filtering. Likewise, there's generally some negative control probes. This very depends on your technology. But probes that shouldn't be present in your sample, if you get a high background there, that will tell you. And yeah, how you would treat those depends on what tech you're working with. But once you've done some QC, you'll then move on to doing some normalization and scaling. So if we consider these 3 cells here, which cell has the highest expression of gene C? Well, they're probably about the same. Normalization corrects for the between cell differences within a gene, so we can compare genes so we can compare cells and groups of cells, so your samples. And scaling is an adjustment across the genes that we use to consider relationships between cells across multiple genes in the next steps, where we want to build that dimensional reduction visualization here. There's a couple of steps in this, but essentially we start with looking at how we're picking out some of the genes that are showing variation between your different cells. You think about a housekeeper that's existing at a consistent level between all of your sample or your cells. It's not going to tell you about the cell types. You want that gene it's expressed really high in like a third of your samples, of your cells. That's going to indicate a cell type. So we pick those genes out. We do a principal component analysis, which sort of summarizes those 200 genes into maybe 10 principal components, just a summary number. And then we summarize that further into these beautiful UMaps for visualizing. So the point of doing those UMaps is this is the fun bit, a fun bit of the analysis, because then you get to plot everything in your experiment. all your QC metrics, all your experimental stuff, what mouse it came from, what your sample was, your groups, your time points, anything you can think of. Because what you're looking for here, an ideal UMAP is probably going to separate by cell type or cell state. And yeah, so we want to see if there's any biases. So you can see here there's a bit of grouping by condition, and oh yeah, this ridge.ident is actually individual samples. So you see a bit of issues. And I'll just skip to the next slide because this shows something that actually occurs reasonably frequently with spatial data. You'll do your UMAP, you'll plot it out, you'll plot out your samples, and you'll notice that your UMAP beautifully separates each individual. And that's not particularly useful for a visualization, because I knew which sample this came from. But there are some methods for batch correction, things like Harmony or RPCA from Surat. But what you do is you take those methods, you tell it which sample was which, and those methods will emphasize or de-emphasize the between sample variation. You can produce a new UMAP, and suddenly you've got all of your samples sort of overplotted on top of each other, but you might have certain biology coming out. Like here, 2 of the samples are actually still from one group, but they're separating out. And, you know, this is where you start to think, well, actually, given my experiment, that is sensible. This is a good visualization for my data. And once you're at that point, you can move on and try and do some clustering because you're using your PCAs, which should be corrected for any batch effects, and you're grouping cells on the basis of their transcriptional similarity. But, you've got, I should mention that, yes, it tends to be there's a lot of maths in doing the clustering, but it tends to be you're just running a command that's like cluster cells. And you might tweak it a couple of times to get more or fewer clusters, but it's generally fairly straightforward. But once you've got that to that point, you kind of want to know what those clusters are. Nobody wants to read about cluster 11. So say this cluster here, we want to figure out what type of cell is in there or what cell state. So this is actually quite a time-consuming part of your analysis, and it is really important because here is you're really starting to pull in that, domain knowledge of, okay, well, this is that type of cell, and that is the basis for your downstream analysis. So you tend to pull in a few bits of information to just make a decision here. The first one, you might look at the expression of genes in a particular group, compare it to the rest of your experiment and say, well, what gene is expressed only in this group? And here you can see, oh, it's enriched for this BCL11B. You might know that that's a marker of a particular cell type. There's certainly databases you can look. Human Protein Atlas is really great for this kind of stuff because they have single cell expression profiles that you can refer to. Even though it's spatial data, the single cell expression profiles are a really good piece of information to make decisions there. Nobody wants to go through that for every single cluster, so you can also use things like a cell typing tool where you get a known reference that you trust, maybe that big experiment, a publication that has a beautiful UMAP with really nice cell types annotated. You go onto the public databases or PubMed, you grab their data, you download their annotations, you use one of these tools and pull their annotations onto your own data set and see, what cell types you see. And lastly, you can also just plot your data spatially. You've got that spatial information, and sometimes that's actually quite, even though you cluster them with no spatial information, you'll see, the epithelial region has a particular cell type. Oh, those are my apical cell epithelia versus the basal epithelia, and it's super obvious, and you can just label it without even looking at the genes. So yeah, you will tend to go from a more broad cell types to maybe a more specific one as you can do. But, bearing in mind, you might be working with a panel. Yeah. So that will get you to this kind of point where you've got your cell types and you know where they are. So yeah, what now? Yeah, now is of course the fun bit where you do the downstream analysis. There's so many options, so many things you can do, but I'll just walk through a couple of sort of examples very quickly. A little bit of a plug here for something, for a collection of how-tos for how to do these kind of analyses. If we consider a experiment where you've got a tumor region, a normal region, and say some T cells, so tumor microenvironment, you could look at the cell type expression between your groups. You don't really need spatial for that, of course, but the data is there. You may as well do it. But what you could then do is, actually compare your T cells in your tumor versus the T cells in the nearby microenvironment. You might be looking at your cancer cells in the immune hot region versus, an immune cold region of the same tumor. Or you might be looking at sort of ligand receptor interaction between two different cell types, and to see if they're co-occurring proximally to each other, and maybe if there's a particular knockout you're looking at, is that sort of relationship maintained? These kind of things. Obviously, there's a lot you can do, but yeah, it's a lot. And yeah, just the spatial data. There's a lot of different toolkits you can use. Here's 3 really good ones. Pick one that makes sense to you. So there's Bioconductor or Surat for the R users, both very good. Python, you've got your SCVerse to choose from. Yeah, you might need to use HPC or a server at certain steps, but a lot of it can be done on a chunky laptop. And yeah, I'll just, in interest of time, I'll just summarize that, yep, you've got all of these steps to do, but you do a lot of, you hit issues at every step, but there's always a way around it. You need to customize your segmentation for your tissue. When you're doing your cell types, you really need to use that domain knowledge and public data to figure out what you've got. And of course, the downstream analysis, we always want more samples, but funding isn't there. So as Michael was saying, it's going to kind of design for your question, so you can answer that. Yeah, so to sum up, yeah, spatial omics is this really exciting technology that can answer these questions that you just can't do with other omics, because it's got that combination of the transcriptomics and spatial information. There's some common processing steps that are shared between most experiments, but once you get to the downstream analysis, it's very much freedom. These technologies are rapidly advancing. Yes, there's no whole transcriptome solution yet, but I'm sure it'll be very, very shortly. At least for the imaging based. Of course, there is for the sequencing based technologies. And yeah, there's different tools for different jobs, and all of these sort of omics work together to answer your biology questions. And yeah, just a sign up link here if you're interested in the hands-on workshop, which we'll be going through that process that I just talked about. Yeah, and some resources, but that'll be on the slides. Yeah, and acknowledgements. So I'm going to stop that here.
Wonderful. Thanks very much, Michael and Sarah. It was a really great a look at the practical questions that are essential to make the best decisions for your experimental design and data analysis. Really valuable. Thanks. So we have some time for questions now. It's the chance for the audience to pop their questions into the Q&A box if you have any, please. Joining us for the Q&A section today, we've had Nicholas Madigan from QCIF volunteer to come along. So you have an extra resource available to you if you'd like to have your questions answered. Welcome, Nicholas. Thanks for being here. So to get us started today, we did have a pre-submitted question, and it was something also that we heard Michael allude to, that spatial omics is very expensive. So I wonder if we could hear how someone should go about designing their experiment to get the most out of the data given its high cost. Perhaps we'll throw to Nicholas. Is that okay to bring you in here, since we haven't heard from you?
Yeah, sure. Yeah, so as Michael and Sarah alluded to, it's about defining your question. You'll have a primary question, and you maybe want to answer a secondary and a tertiary question as well, because you're doing this technology. But if that is at the cost of your primary question, then you may need to rethink, the answers or the amount of samples that you are going to do. In that context, what I'm meaning by that is that, if you're choosing an extra subtype of cancer because you want to maybe know what that is, but then you're using less samples from your primary cancer, you may not have the statistical power to really nail down what you're after. So you might have to put all your eggs in that one basket to properly answer that question. So
Great, thank you. Did Michael, you want to follow up with anything?
I think another thing you can consider is combining different omics technologies. So a common workflow can be to use a technology like single cell sequencing to first do the more high throughput exploratory data analysis, and that lets you get a bit more bang for your buck in that regard. And then spatial can, the more expensive spatial technology can be used for a more refined or targeted analysis. So that's another way you can tackle that.
Great, thank you. We have a question that's come in from today's audience about asking if anyone has any comments on superimposing different types of spatial omics data. For example, MS-based imaging, we're talking proteomics or metabolomics, and transcriptomic imaging.
Yeah, I can probably comment on that.
Thanks.
Yeah, so I haven't personally done any multi-omic spatial myself, but I know that it is definitely something a lot of people are looking at. So the software packages do definitely have support for this kind of thing, including like multiple assays sort of on the same cells. From what I can gather, one of the main issues is kind of the registration of the images in image-based stuff to get them lined up. And I think certain technology, I think I have heard of people doing that with cosmics as well, on the actual same samples, doing a protein and a RNA assay. So yeah, I think it's exciting.
Great, thank you. We've got another question here. It's a kind of general question open to anyone. How advanced is spatial metabolomics? Any thoughts on that?
I have not had any experience with spatial metabolomics, so I don't know if anyone might have a better answer.
I haven't seen any either, but yeah, like I think people are working on it, and there are some data sets out there, but I think for most of the different omics and what you can think of it's and if the machines can do them, you fall into the same issues that Michael explained with the different technologies. Either you can't do too many, or you can't do single cell. So I think you end up in that same kind of realm with whatever omics. So it's as long as the machines can read them and barcode them, then it can be done.
Yeah,
okay, thanks.
Yeah, sorry.
Go, Michael.
I was just going to say, I think in general, I too have not dealt with metabolomic data, but I think for the most part, the analysis would be somewhat of a similar process, although I think maybe some of the underlying statistics might be a little different because you're dealing with continuous data versus counts, but ultimately, yeah, it would be fairly like the same level of advanced. Yeah.
Mm-hmm.
I guess it depends a lot how many metabolites the technologies can measure at the same time.
How about we swing to the other end of the pendulum then? And can you tell us what are the kind of research questions that are the bread and butter? What are you seeing most often? What are the most common sort of biological questions that are being answered?
It's quite popular in cancer microenvironment, I say, is just because having that ability to, see, all right, that bit's the cancer and that bit's nearby, and because it's so important, like,
Yeah.
The,
Around what sorry, Margaret, particularly around what cells are signaling to each other. If I have, these cells in the tumor microenvironment, do they have receptors or on them that would receive signals from immune cells on the outside? What signaling is going on, and then how the idea behind that is then to, what drugs can we use to treat these cancers if it's expressed on the outside of a cell. Then maybe we can design targets, design drugs to target those cells specifically.
Mm-hmm. Would anyone like to talk about how AI is going to influence the analysis of spatial omics data? Where are we going?
I was already doing the I think the segmentations methods are already AI based. It's an image stuff, which is the bread and butter of it.
Mm-hmm. Do you have a feeling for what's the exciting directions that it could go into?
Yeah, I think the one of the exciting areas is the digital pathology, field, where the spatial omics experiment is expensive and time consuming, but we could use just a simple $1 H&E stain, which is done with every spatial omics, then to know that cell sitting here next to these ones is this type, and we can infer that from the cheaper slide that's in every pathology lab that's out there. So so to use all this, you don't need to go off and do the whole transcriptome, metabolome, or whatever ohm to be able to then interpret a pathology slide, cheaply. So that's one of the areas, and the AI is already being used in the cell type annotation as well. So there's several packages out there that are using that, using all the transcriptome data to compare with yours.
Alrighty. Well, thank you very much for joining us today. We'll need to leave it here given our time constraints, but I'd like to once again express our gratitude to Michael and Sarah for their presentation today, and also for Nicholas for joining us to answer questions. Thanks so much. So you heard during the talk as well that there's an opportunity to learn more about analysing spatial omics, and you can do this by joining a workshop that's coming up in October. There are spaces on this workshop available now, but they are limited and applications close on the 10th of October. So you can find out more about this workshop via the links you've been provided, and it's also available on our website, of course. So to wrap up today, we'd like to acknowledge our funding. The Australian BioCommons is enabled by NCRES via Bioplatforms Australia. And we hope you'll consider joining us again for our next webinar. Wonderful to see everyone here today. We hope you've enjoyed our presentation, and we look forward to welcoming you again in the future. Thanks so much. Bye.